Okay, welcome back to the Classics of Immunology Journal Club. And before we get started, don't forget to like this video if you do, and click on the bell to get notified, and scroll down onto the website, subscribe to the YouTube site, and scroll down on the site, and you'll find my website myself. And then on the website, you can check on the writing tab, and you can find all the classic papers of immunology to educate yourself. And this is open to non-immunologists too. You can become proficient in immunology if you do the historical approach to the field. I've noticed in reading these papers, that it's very, it's very difficult to read a paper and to figure out the chronology of the progression of the science in terms of not only what was done and when it was done, but who did it. That's all scrubbed, particularly from the last 50 years of scientific publication in every field you look at. The third person is used a lot. This happened and then that happened and so forth. And you get the idea that nobody really did anything. It just happened. And so you have to really check out the people, find out who they are, Google them, and go from there. The paper we're going to review today was published in Science in January of 1991. And the authors are Jacques Banchereau, Paolo de Paoli, Alain Valle, Eric Garcia, and Francoise Rousset. And the title of this paper is long-term human B cell lines dependent on Interluekin-4 and antibody to CD40. Now, the background on this paper is that Banchereau was an investigator with the Schering-Plough lab for immunological research that was present in an outfit that Schering-Plough, the large pharmaceutical company, established just outside of Lyon in France. And that's why we're mostly continental investigators in this whole thing. The background of this whole business of trying to get B cells to of course, grow and continuously grow. You can imagine if you could do that, you wouldn't have to make monoclonal antibodies anymore. You could just have normal B cells making your monoclonal antibodies. You wouldn't have to do this fusion thing and all the selection and so forth and so on. Now, this rests on the idea that was published by Av Mitchison from London back in 1971. So this is 1991. 20 years later, and I've made the astute observation that in order to get really good T cell help to B cells so that they knew what to do, there had to be some physical contact between the T cell and the B cell, which was called cognate recognition. In other words, they had to kiss one another. And that's interesting. And just as an aside, once interleukins were into this fray, I figured that this cognate recognition was not really true. But this paper maybe will convince you that it is. Now, it turns out that one of Av Mitchison's postdocs was a fellow by the name of Ed Clark, who was an American, and he ended up in Seattle at the University of Washington. And in 1986, he was looking for monoclonal antibodies that would be useful in the interrogation of how B cells and T cells interact and so forth and so on. And he came up with an antibody that ultimately was found to recognize one of the CD molecules, or it was named one of the CD molecules after it was recognized by this monoclonal antibody, and it was called CD40. You can remember CD3 is the signaling component of the T cell receptor. CD4 marks helper cells and CD8 marks killer cells and so forth. Ed Clark back in Seattle characterized this antibody and so forth and found that it would augment the proliferation of B cells. It was thrown in with interleukins like Interleukin-4 and so forth and so on. But nobody had really taken advantage of these findings, these recent findings in B cell biology, to try to recreate what the T cell people had done a decade earlier, essentially. And the introduction to this paper, they go into a little bit into the history, and they point out that T cell lines, antigen-specific functional T cell lines, and T cell clones had been available for a whole decade, but nobody had made long-term B cell cultures. I should have said, I forgot to mention that once they got the antibody to this CD40, they found that it was reactive to a cell surface molecule on B cells that fell into the TNF receptor family. So this was a receptor molecule that could cause growth and other functions in these kinds of cells. They found in these experiments that Ed Clark and others had done was that it was critical that you had antibodies to trigger the CD40 surface TNF-like receptor molecule. In this paper, they set out to try to create long-term B cell lines. So they developed a B cell proliferation assay to quantify that, and they used B cells from human tonsils. Remember, this is the long-term human. The title of this paper is long-term human B cell lines, which is sort of not the usual fare for these cellular immunologists back in those days. And so they would take human tonsils that had a lot of B cell follicles, and they'd purify the B cells from these tonsils after they got them into a single cell suspension, and they could partially purify them over a Percol density gradient centrifugation. And they found that they took another chapter from the T cell people. The T cell people had found if they used mouse fibroblasts that had cloned into them a human FC receptor molecule, FC gamma R2, that would bind the monoclonal antibodies via the FC fragment of the antibody and then antigen binding regions and immobilize them in such a way that it was a much better stimulus than if they used try to use either soluble anti-CD40 or anti-CD40 bound to plastics. And so they tested several different kinds of antibodies to develop their assay and so forth. And they found that the monoclonal antibodies reacted to CD40, and they had two at this stage, would cause the cells to proliferate, and they measured them in a 3-day culture and then a pulse of tritiated thymidine. They looked at also antibodies to CD19, CD21, CD23, 24, and 37, and none of them worked. And if they added in interleukin-4 together with their anti-CD40 antibodies, it caused an enhancement of marked proliferation. So if we go to the results and look at these data in terms of the screen sharing, the first thing I want to draw your attention to is table 1. And here's where they tested these monoclonal antibodies over here, the different lists of them. And this is what they recognized on the surface of cells. This was the CD40, these first two, and then 19, 21, and so forth. As I said, this is then thymidine incorporation into the proliferating B cells. And if there was no monoclonal antibody there, they had a very low incorporation of thymidine. And then with the CD40, anti-CD40s, they were markedly positive, whereas all the rest of the monoclonal antibodies that they were able to find and test were negative. And then if you look at figure 1 here, this is now listed on the ordinate here, the tritiated thymidine incorporation, and this is time in days where they would pulse the different cultures with thymidine. And if you look at the different symbols here, triangles down here are interleukin-4, antibody interleukin-4 alone, and then the circles, or no, these are squares, I think, and that's the anti-CD40, and that gives a decent thymidine incorporation. But here's the two together, anti-CD40 plus interleukin-4, and you can see this marked enhancement. Then if you look at figure 2, it's the same sort of thing, except now we've got the number of cells, the cell concentration in both of these, both A and B in terms of these figures. And this, which markedly stands out, is anti-CD40 plus interleukin-4 that was present during all the days of this culture that went out to 13 days, essentially. This is anti-CD40 alone. And down here is no addition. So they just go down and stay stable and then die off. In the lower panel B, You see that this is the cumulative cell concentration calculated from when they every several days they would split the cultures down and then allow them to grow up again and then split them down again. And that's how they derive these data in terms of the cumulative accumulation, putative accumulation of cells that they might be able to grow. If they took them out of their stimulant, then they immediately dropped off. Then figure 3 is not a good thing, but basically it's an immunohistochemical stain for EB virus, because everybody knew that a long-term B cell proliferation could be activated by Epstein-Barr virus infection of B cells, and these were negative, essentially. This is then a frequency analysis and of the number of cells responding in these cultures, and it was essentially 1 out of 3, or 1/3 of the cells. And so there was a sizable fraction of the cells that were responding in this particular series of experiments. The importance of these whole things is that in the discussion, they said that these findings would allow people to make factor-dependent human B cell lines. And they speculated whether the CD40 molecule, because it looked like a TNF receptor family member, it could be a receptor for a cytokine that yet had not been discovered yet, or it could be a receptor for a cell surface ligand that was, that might be participating in this so-called cognate recognition when the T cell kisses the B cell. And they left that up for conjecture at this stage. And they also wondered whether or not you could use this system to first select for antigen reaction and then put the cells into the anti-CD40 on the one hand, plus the interleukin-4, and they might be able to duplicate what we did in the T cell story with just using interleukin-2, but we would add that after we selected for antigen specificity. And they also thought that this ought to be useful to study B cell activation and to really try to find out what the different signals were or are that go into this whole phenomenon of T cell help. So it turns out that although they showed in this paper that you could grow T cells for quite some time, I think they had one series of data that would go up for a month or two, essentially. Life is never really all that easy, because what they subsequently found after this paper was published in Science and so forth and so on was that the cells would stop growing after a few weeks and so forth. They would terminally differentiate and become plasma cells and or memory B cells, which is good. I mean, if you want to have, that's what B cells do for a living, but they couldn't create their long-term B cell lines. And so looking forward, that was 1991, that was 30 years ago. And even now today, if you trace back into the literature up to the present time, and actually I've got a couple of papers here that I wanted to draw your attention to. One investigator who's been into this field for a long time is Garnett Kelso from Duke University, and he has a paper out in August of 21 on the continuous culture of mouse primary B lymphocytes by forced expression of the transcription factor BOC2. And so this is in the Journal of Immunology in August 13th. So you might want to take a look at that. And there's another paper that he drew my attention to that is out in Immunity in February 8th, 2022. So up to the present time, it's not clear to me whether we really know what all this business is of what T cell help really entails at the molecular level. And that's important, because if we can pin down what all the proliferation physiological factors are and all of the differentiation factors that are important for B cells. I should point out that T cells don't differentiate like B cells, because B cells undergo what they call class switch recombination, where they switch their heavy chain, and we went into this a little bit with interleukin-4 from the mu heavy chain to IgG, A or E. And so class switch recombination is an important differentiative phenomenon that is, I've got a lot of people studying that. And then it also gets somatic cell hypermutation, affinity maturation of the antigen binding site of the B cell. The T cells don't do that. They don't do really either one of those things. T cells are, I hate to call them a little simpler, but B cells, because they're so complicated, they need a lot of T cell help. And I think that as immunology goes on, the more we find out how all this works at the mechanistic and molecular level, we can use that information to manipulate whatever we would like the B cells to do in a therapeutic fashion for autoimmune diseases, for example, and so forth. So I'll stop there. Don't forget to like the video and click on the bell and so forth. And tell all your friends, and then come back next week. Thanks so much. It's been fun.